Hello, everyone. Thank you for joining me today. In this presentation, I'll be walking you through the various ways and best practices for DNA and RNA extraction and purification. DNA is a double-stranded molecule that carries the genetic blueprint of living organisms. It contains instructions for building proteins and is responsible for heredity, determining traits passed from parents to offspring. DNA is found mainly in the cell nucleus. RNA is a single-stranded molecule that plays a key role in gene expression and protein synthesis. It acts as a messenger carrying genetic information from DNA to the ribosome and also has structural and catalytic functions in the cell. As you know, obtaining high-quality nucleic acids is the foundation for many downstream applications such as PCR, qPCR, sequencing, and diagnostics. Let's begin with a general overview before diving into specific methods. Regardless of the method used, nucleic acid extraction typically involves 4 key steps: lysis, binding, washing, and elution. The goal is to efficiently lyse the cells, bind nucleic acid selectively, remove all contaminants, and elute pure DNA or RNA. The choice of method depends heavily on your sample type and the downstream application. Let's now look at what kinds of samples we commonly work with and what factors we need to consider. Nucleic acids can be extracted from a wide variety of sources: blood, tissue, cultured cells, bacteria, plants, even viruses. Each sample type poses unique challenges, such as high levels of polysaccharides in plant tissues or RNases in blood. To ensure successful extraction, we must consider sample freshness, storage conditions, and potential inhibitors. With that in mind, let's take a look at the most commonly used extraction methods available. There are several main strategies for DNA/RNA extraction. These include organic extraction with phenol chloroform, silica column-based methods, anion exchange chromatography, magnetic bead-based systems, and TRISol or CTAB-based protocols for specialized samples like plants or RNA. Each method has its own pros and cons in terms of yield, purity, ease of use, and scalability. Let's start by revisiting the traditional organic extraction method. This method uses phenol and chloroform to separate nucleic acids from proteins and other cell components. It offers high yields and is quite effective for tough samples. However, the process is labor-intensive, requires hazardous chemicals, and is not suitable for automation. Given these limitations, many laboratories now prefer safer and faster methods. One such alternative is silica column-based extraction. In this approach, DNA or RNA binds to a silica membrane in the presence of chiotropic salts. This is widely used in commercial kits due to its speed and simplicity. While it's effective for many applications, it can struggle with viscous samples and is less scalable for high-throughput needs. That brings us to a more modern and versatile method: magnetic bead-based extraction. Now, let's take a closer look at another common technique for DNA purification: anion exchange chromatography. This method relies on the interaction between positively charged defunctionalized resins and the negatively charged phosphate backbone of DNA. Under carefully controlled pH and salt conditions, DNA in the sample binds to the resin. After binding, a series of stringent washing steps remove impurities such as proteins and cellular debris. Finally, DNA is selectively eluted by changing the salt concentration. This method is particularly valued for producing high-purity DNA, making it ideal for sensitive downstream applications like transfection and DNA sequencing. However, one downside is the higher cost of the resins, which may limit its use in routine or large-scale extractions. Magnetic bead-based extraction has rapidly become the preferred method in both research and clinical labs. Here, functionalized magnetic beads bind nucleic acids under specific buffer conditions. By using a magnet, the beads and bound nucleic acids are easily separated from contaminants. The process is highly adaptable, allowing automation and consistent results. Let's explore why this method is gaining popularity. This method offers many advantages. It supports high-throughput workflows, reduces cross-contamination, and is gentler on samples compared to spin columns. The method is scalable and automation-friendly, making it ideal for labs processing many samples or integrating with downstream workflows. It's especially useful for complex or difficult matrices such as blood, stool, or environmental samples. Next, I'll share some useful tips to get the best performance out of magnetic bead-based extractions. To maximize your yield and purity with magnetic beads, make sure the beads are well resuspended before use. Ensure complete lysis of your sample. Allow adequate binding time. Use enough washing buffer to remove impurities. And elute in a low-salt, nuclease-free buffer. These steps help prevent loss of nucleic acids and avoid carryover of inhibitors. Let's move on to some additional tips for RNA-specific extraction. RNA is much more sensitive than DNA due to the ubiquitous presence of RNases. Always wear gloves, use RNase-free consumables, and treat surfaces or solutions with DPC when needed. Use lysis buffers containing guanidinium isothiocyanate or beta-mercaptoethanol to denature RNases. For clean RNA, include a denase treatment step. Finally, store RNA at -80 degrees Celsius in ethanol or an RNase inhibitor solution for long-term stability. Next, let's briefly review tips for handling DNA samples. For DNA, it's important to avoid mechanical shearing, so pipette gently and minimize vortexing. Avoid over-drying DNA pellets, which can make them hard to dissolve. Treat with RNase if you need DNA free from RNA contamination. Check your purity with A260/A280 and A260/A230 ratios to ensure you're working with high-quality DNA. But what if something goes wrong? Let's talk about troubleshooting. When you face problems like low yield, degraded RNA, or poor purity, here are some common causes and solutions. Low yield? Check your lysis efficiency and binding time. Degraded RNA? You may have RNase contamination. Double-check your materials. Poor purity? It might be due to insufficient washing. Add extra wash steps. Bead clumping? Make sure the beads are thoroughly resuspended. With consistent technique and attention to detail, most of these issues are avoidable. Now, how do we assess the quality of the nucleic acids we extract? Assessing nucleic acid quality is crucial before moving to downstream applications. Spectrophotometry helps you quickly assess purity. You should aim for A260/A280 ratios of 1.8 for DNA and 2.0 for RNA. Fluorometric methods like qubit are more sensitive and accurate for quantifying low concentrations. For integrity, run a gel or use a bioanalyzer. For RNA, sharp 28S and 18S bands indicate good quality. Next, I'll highlight some real-world applications and case studies. Here are just a few examples where optimized extraction methods made a real difference. During the COVID-19 pandemic, magnetic bead-based RNA extraction enabled fast, high-volume testing. In plant research, CTAB followed by bead purification gave clean DNA for genome sequencing. In clinical diagnostics, automated magnetic platforms ensure consistent DNA prep from blood samples. These examples highlight how method choice impacts efficiency and data quality. To summarize, high-quality nucleic acid extraction is key to success in molecular biology. Method selection depends on sample type, throughput needs, and downstream applications. Magnetic bead-based methods offer superior flexibility, scalability, and purity. With proper technique, troubleshooting, and quality control, you can consistently obtain excellent results. Thank you for watching so far. For more information about CD bioparticles, magnetic silica particles, ion exchange chromatography resins, and magnetic bead-based DNA and RNA extraction and purification kits, please visit cd-bioparticles.com.